Hi everyone, welcome back to my A-level biology lecture series. In this lesson, we will explore the structure and the function of chloroplast. At the end of this lesson, you will be able to describe the relationship between the structure of chloroplast as shown in a diagram and electron micrograph and their function, as well as state that within the chloroplast, the thylakoids, including thylakoid membranes and thylakoid space, which occur in stack called grana, are the sites for the light-dependent stage, and stroma is the site of the light-independent stage. All right, let's get started. Before we get into the lesson on chloroplast, let's quickly go over some background on adaptation of the leaf for photosynthesis. So the leaf is a complex organ composed of multiple tissues structured to maximize photosynthesis. So the dicot leaf blade consists of upper and lower epidermis. Both layers of epidermis form a protective outer layer. Palisade mesophyll and spongy mesophyll are the tissues specialized for the light absorption as well as the gas exchange. Vascular bundle includes xylem and phloem, which are responsible for water and nutrients transportation. Let us look at this, the histology of a dicot leaf lamella. So first of all, upper epidermis, which form a protective layer, lower epidermis, which includes a specialized cells known as the gut cells. So the gut cells control the stomatal opening. Singular stoma, plural stomata. The next layer is palisade mesophyll, where this layer of cells carry out photosynthesis with lots of these chloroplasts inside the cells. Spongy mesophyll, which are irregular cells that form a loosely packed tissues. So within this layer of spongy mesophyll, due to the loosely packed nature of the cells, we can actually find this substomatal cavity or substomatal air spaces. So vascular bundle consists of xylem and phloem. As we have learned in AS topic, xylem transports water and mineral, while phloem carry out translocations of the assimilate. So both phloem and xylem form vascular bundle. Let us have a summary on different tissues of a dicot leaf. Let's start with the upper epidermis. First of all, the cells are thin, flat, and transparent. And this is very important to allow the light to pass through the upper epidermal cell so that the light energy can penetrate through this layer and reach the mesophyll layer. At the same time, they have this waxy transparent cuticle, and they reduce this waxy cuticle reduces the water loss from the plant through the cuticular transpiration. The next layer, lower epidermis. So this layer contains many stomata, each bounded by two gut cells. So the gut cell regulates the diameter of each stoma, and the opening of the stoma allows the gaseous exchange, enabling the net diffusion of oxygen out of the leaf and carbon dioxide into the leaf during photosynthesis. So please bear in mind that when we talk about gaseous exchange in plants, we need to mention during photosynthesis or during respiration, because during photosynthesis, there is a net diffusion of oxygen out of the leaf and CO2 into the leaf. However, during respiration, there is a net diffusion of oxygen into the leaf and the net diffusion of CO2 out of the leaf. So please take note. The next layer, let us look at the palisade mesophyll, which is the most important layer or important tissues when we study chloroplast as well as photosynthesis. So what are the features of the palisade mesophyll enable them to adapt for photosynthesis? First of all, they are a long cylindrical cells arranged at the right angle, or we say perpendicular to the upper epidermis. So being a long cylindrical cells at the right angle definitely reduce the number of light absorbing crosswalk in the upper parts of the leaf. You try to imagine that each leaf must maintain a particular thickness. So if these palisade mesophyll are arranged parallelly to the upper epidermis, therefore it must be multiple layer of the cells. So if there are multiple layers, each layer of the cells will absorb the light. So means that the lower layer will not have sufficient light energy for them to carry out photosynthesis. So in order to reduce the number of layers of the cells of this palisade mesophyll, so the arrangement of the cells is special. So the cells are arranged perpendicularly to the upper epidermis, reduce the number of layers of the cells, and at the same time reduce the number of the light absorbing crosswall, the cell wall in the upper parts of the leaf. And because they are cylindrical cells, tightly packed, okay, so this will create narrow air spaces between the cells. You try to imagine that if they are round, so when you tie all these round shaped cells together, there are air spaces in between the cells. So therefore, with these narrow air spaces between the cells, it provides a large surface area for contact between the palisade mesophyll cells and the air. Third, if you look at the cells, the wall is very, very thin. So definitely provide a short diffusion distance for the gas exchange, where CO2 enter into the cells by diffusion and O2 out of the cells by diffusions, okay? Hence increasing the rate of gas exchange during photosynthesis. Next, the cells contain a large central vacuole surrounded by thin peripheral layer of the cytoplasm. Because of this large centrally located vacuole pushes the cytoplasm near to the cell wall. Therefore, chloroplasts actually are located near to the cell wall. So this again reducing diffusion distance and enhance the rate of the gas exchange during photosynthesis, where CO2 diffuses from the surrounding air spaces into the mesophyll cells and then into the chloroplast stroma, for example. And at the same time for O2 to diffuse out of the chloroplast and then to the air spaces. Finally, we have these features where the cells have a higher number of chloroplasts and these chloroplasts can move within the cells. Having high number of chloroplasts allow maximum absorption of light energy, and the movement of chloroplasts away from excessive light intensity actually protect them from damage. The next layer that we are going to study here is spongy mesophyll. So they are irregular shaped cells, loosely packed with large air spaces. So with these large air spaces actually provides an uninterrupted diffusion pathway for the exchange of the gas between the atmosphere and the palisade mesophyll cells, because these loosely packed cells create the air spaces. So these air spaces actually allow the diffusion of the gas from the atmosphere through into the air spaces through the opening stomata. Lastly, let us look at the vascular bundle, which consists of xylem tissues and phloem tissues, as well as fiber. So xylem, as we know, that transport water and the dissolved mineral ions from the root to the shoots, and phloem Okay, sieve element transport water-soluble assimilate from the source to the seed. Then the fiber actually provides structural support for the plant. So now let us look at the chloroplast structure as well as its functions in photosynthesis. So in terms of the chloroplast structures, they are lens-shaped, okay, biconvex, approximately 3 to 6 micrometer in length. So they are found in the photosynthetic leaf and other photosynthetic organs of the plant as well as in algae. So therefore, not all plant cells have chloroplast. Okay, for example, the root hair cells, cortical cells, the cells at the stem, they do not have they do not carry out photosynthesis, therefore they do not have chloroplast. So in terms of membranous system of the chloroplast, divide chloroplast space into the 3 main compartments. The first compartment is known as the intermembrane space, formed between the outer membranes and inner membranes of the chloroplast. So therefore, chloroplast is a double membrane bound organelle. Chloroplast stroma, okay, and enclosed by the chloroplast inner membrane, as well as the thylakoid space enclosed by the thylakoid membrane. So the content of chloroplasts are separated from the cytosol by an envelope made up of 2 membranes separated by a very narrow intermembrane space. When we use the word envelope, the envelope actually represents 2 membranes, the outer membrane as well as the inner membrane. Inside the chloroplast is another membranous system consists of flattened interconnected sacs known as thylakoids. And in some regions, these thylakoids are stacked together. So each stack is known as granum, and plural is known as grana. So the fluid outside the thylakoids, okay, surrounding the thylakoids is known as a stroma, or more precisely, we call them as chloroplast stroma. Please be careful when you spell or when you write the word stroma, avoid, okay, or be careful, do not misspell it as stoma, okay? Stoma is the opening of the lower epidermis where it allows the gas to diffuse in and out of the leaf. So this stroma contains circular chloroplast DNA, 70S ribosome, starch granule, as well as many enzymes that catalyze the light-independent reaction, which is Calvin cycle. We are going to have a lesson focused on Calvin cycle soon. And this compartmentalization enable the chloroplast to convert the light energy into chemical potential energy during photosynthesis. Let us look at the structure of chloroplast. First of all, outer membrane, inner membrane, and the space between these 2 membranes is known as the intermembrane space. So a stack of thylakoids, okay, form a granum. Let us zoom into the structure of a granum, where you can see that it's formed by this thylakoid membrane, and the space is known as a thylakoid space. So thylakoid membrane and thylakoid space form thylakoid. There is a region known as a lamella. Lamella is the unstacked thylakoids that connect grana together, forming the interconnecting system. The semi-fluid structure that's surrounding this grana is known as chloroplast stroma. Within this chloroplast stroma, we can find circular chloroplast DNA, 70S ribosome, as well as a starch granule. In fact, the chloroplast stroma is quite similar to the mitochondrial matrix, where inside the mitochondrial matrix, 70S ribosome and circular DNA can be found. So let us focus on each structure and what is their features as well as their function. Let us look at the envelope, consists of outer and inner membrane. So it consists of phospholipids bilayer containing transport protein. And this phospholipid bilayer separates the internal chloroplast environment from the cytoplasm. And these two layers of membrane permeable to small molecules such as carbon dioxide, water, ADP, and so on. And this is very important to allow the entry of the essential molecule for photosynthesis. The next regions or structures, let's look at the grana. So grana is formed by a stack, by the other stacks of thylakoids. So due to this, the stack of thylakoids increases the surface area, for light absorption during the light-dependent reaction, as well as to increase the rate of ATP synthesis through photophosphorylation. As we have learned in previous topic, ATP can be synthesized through three ways. The first way is known as a substrate-linked phosphorylation. Second way, during oxidative phosphorylation. Both ways are found in the respiration. And the third way where ATP can be synthesized is through photophosphorylations during photosynthesis, or more precisely, during light-dependent reaction. Thylakoid membrane contain photosynthetic pigment, electron carrier, as well as ATP synthase. So during light-dependent reactions, light energy absorbed by this photosynthetic pigment, particularly chlorophyll, and the electron transport chain, ETC, transfer photoexcited electron through a series of carrier, and ATP as well as reduced NADP are produced, which both of them are required for the Calvin cycle, which is a light-independent reaction. And Calvin cycle, the role of Calvin cycle is to synthesize organic carbon compound. Next, thylakoid space. It is an enclosed fluid-filled compartment with low pH or high proton concentration. So this thylakoid space allows accumulations of proton to generate a steep proton concentration gradient between chloroplast stroma and the thylakoid space. Thylakoid space contains enzyme for photolysis of water molecule. This process is very important to split water molecule into the hydrogen ions, proton, and oxygen. During this process, electrons is released. The electrons is involved in the photosynthesis. Lamella. So it is a flat, unstacked membrane sheet that connects the grana together. So basically, lamella link grana stacks forming a continuous thylakoid membrane space, and it acts as a site of cyclic photophosphorylation to produce ATP only. Chloroplast stroma contains circular DNA as well as 70S ribosome. So with the circular DNA that contains the gene, this allows transcriptions to take place, as well as the translation occur in 70S ribosome to synthesize chloroplast protein and enzyme. In the stroma, the enzyme for Calvin cycle can be found. So this enzyme catalyzes the key stages of Calvin cycle to produce organic carbon compound. As well as the DNA, 70S ribosome, and enzyme, the structure of chloroplast stroma, which is colorless and transparent, which allows the light to reach the thylakoid membrane to reduce the absorption of the light energy. And there are starch granules found in the chloroplast stroma as well. So they are the dense and insoluble carbohydrates granule. They are the temporary storage of the excess glucose produced during photosynthesis. At this moment, you may find those functions and the features quite complicated because we haven't done the reactions, the photosynthesis processes, as well as the reaction. Once we have done the whole series about the photosynthesis, which include the light-dependent and light-independent process, then we can come back to this lesson again to revise, and you will be able to get the concept more or clearer when you have done the photosynthesis reaction. This electron micrograph or transmission electron micrograph show the features of a chloroplast, where you can see that there are many stack of thylakoids that form grana, and these grana are interconnected by the lamella. So in our syllabus, you are required to identify the structure of components of a chloroplast. And with that, we have come to the end of this lesson. Hope you found it helpful and easy to follow. If you have any question, feel free to drop them in the comment section. See you in the next lesson and have a great day.